So the session today, we're talking about rapid transformations of forests that, that they're experiencing as a result of changing climate, disturbance regimes, human pressures, and there's been a bunch of recent conceptual advances, really highlighting the importance of tree demography. So regeneration, growth, mortality, shaping overall forest responses to these global change drivers. And understanding these demographic processes on large scales requires an enormous amount of data and depth, breadth, different domains, and really well suited to national inventory programs. And so this session is really going to highlight some research that brings forest inventory data to bear on multiple aspects of tree demography to understand how global change influences forest ecosystem trajectories across different ecological scales. So we got 90 minutes today, 6 presentations. You all probably know the story now, 15 minutes a talk. Gonna hold questions till the end of each session, hopefully time permitted for some questions. You've probably heard the routine about things like photographs, screenshots, all that stuff without presenter express verbal or written consent. And any audience members, if you're having technical issues, you can direct a message to the moderators. We'll try to help out. So our first presenter is Ayanna St. Rose from Oregon State University, who is going to be presenting on post-fire regeneration in the Pacific Northwest. So please take it away.
Hi, everyone. My name is Ayanna. Like Harold said, I'm a postdoc working with the Landscape Ecology Modeling, Mapping, and Analysis group out of Oregon State University. And I am currently working on understanding post-fire seedling regeneration by looking at factors affecting tree seedling abundance and composition in the Pacific Northwest. Today, I will be talking a little bit more about the analysis approach that I am taking and going over some preliminary results. And at the end, I would appreciate some feedback and comments based on that. So the Pacific Northwest houses some of the highest concentrations of carbon in the United States and holds great ecological, cultural, and economic value. But it is facing growing challenges because of an increase in the frequency and intensity of wildfires, which can be attributed to compounded climate and anthropogenic factors. Because the long-term resilience of these forests depend on the capacity to regenerate, post-fire seedling regeneration is critical to maintaining these ecosystems, especially in the face of climate change. Assessing post-fire seedling regeneration is complex as it relies on vulnerable stages like the availability of a viable seed source, successful germination, and survival to the sapling stage. And many factors come into play here, like climate, terrain, canopy structure, and so on. And further complexity is added because different species respond differently to each of these factors. A focus on the correlation and magnitude of importance of these factors in research has yielded valuable insight, but also highlights a unique gap in understanding the complex interactions between various drivers of seedling regeneration and how these interactions vary. So to address this gap, we asked 3 questions. What are the direct, indirect, and combined drivers of seedling abundance and composition? How do these drivers differ before and after fire events, and in the case of repeated fires? And what is the probability of seedling presence given these drivers? Our team generated various layers of environment, structure, and soil, and we also sourced fuzzed seedling and tree abundance data from FIA. We separated that data into no fire and plots that were affected by fires and did some preliminary data exploration with correlation analyses. We've started running our structural equation models to examine various pathways affecting species abundance, and given these outputs, we plan to run generalized linear models to predict seedling incidence and run non-metric multidimensional scaling to examine the variation in community composition. This talk is mostly focused on the application of the structural equation model and to better understand seedling regeneration. So structural equation modeling is a statistical technique that evaluates complex relationships between variables by combining factor analysis, multiple regression, and path analysis. So to better understand these concepts, let's take a look at our hypothetical path analysis, which we tested. So in structural equation modeling, there are concepts of direct drivers, which are those drivers that would have a direct impact on seedling regeneration. There are indirect drivers, which are those drivers that would have an influence on seedling regeneration through a more nuanced way. And we have combined effects when we see both of these factors interacting. So here, for example, disturbance is our latent variable, and you can think of a latent variable as a common factor or a common grouping. And disturbance, hypothetically speaking, is measured by fire intensity, fire frequency, and time since disturbance. And disturbance has a mediated effect on seedling abundance through affecting canopy complexity. For our analysis, we will focus on Oregon, California, and Washington, but today we hone in on Oregon plots. We did this analysis using lodgepole pine as our species because it is one of the more abundant species in our dataset. So there are over 1,500 plots where lodgepole pine can be found, and out of those plots, 261 of those plots were burnt. Among these burnt plots, we can see that the number of plots generally decreases as the time since fire increases in each of the ecoregions that we looked at. So for our examination of the data using correlation analysis, we start to see some of the relationships to expect while running the structural equation model. For example, abundance was positively correlated with elevation, and that makes sense because lodgepole pine species are generally found at higher elevations. And we can also see that canopy cover negatively affects abundance, which also makes sense because this is a shade-tolerant species. As preparation for the structural equation model, we ran a confirmatory factor analysis to test our hypothesized model by confirming the relationships between observed variables and their underlying latent constructs. So here we can see the significant groupings with the significant correlations between latent constructs. We must note that we lost some measured variables. So for example, we lost the number of fires variable that would be included in disturbance, and we also lost the porosity variable that was included in our soil latent variable. So we can also see the strength of the correlation relationships between the measured variables and our latent variables. Generally, the latent factors had a significant effect on seedling abundance. We see that here terrain is having both a mediated and direct effect on abundance. And while disturbance and competition are having a direct effect on seedling abundance. But we should take this with a grain of salt because we see that we have a high root mean squared error of association as well as a low fit indices. So this kind of tells us that we might be missing some important pathways or predictors in our model. So this is also further emphasized by the fact that we had a very low amount of variance in our seedling abundance being predicted by our model. So in summary, predicting abundance is complex and involves many direct, indirect, and combined effects. We were able to successfully decipher latent variables and their relationships to each other and how they relate to lodgepole pine seedling abundance. But our model did not do a good job at explaining all the variation in lodgepole pine abundance. So we said that this could be because error was introduced at different points of our model. For example, we used first plots. So maybe if we use actual location of the plots, we would limit some of that error. And two, we could also include more variables because there may be some processes and local factors that we are not capturing with the variables that we already have. So next steps, we'll be going back to the drawing board and hopefully adding more variables, tweaking the model so we could move forward with the remaining parts of our workflow, generating generalized linear models and our NMDS. And we plan to extend this analysis to multiple species as well as plots in California and in Washington. So with that, I want to say thank you to my collaborators on this work, Matt, Cameron, Erin, David, everyone present here and the people of FIA and my group in Oregon State University. Okay. Any questions?
Great. Thank you very much. We do have a couple minutes for questions.
Awesome. Or comments. I'm open to comments because this is really, it's a really new, well, it's not really new, but it's developing. It's still developing. I see Daniel with his hand up.
Yeah, I have that was a great talk, Ayanna. Thanks. This is something that I've grappled with myself trying to think about regeneration is how seedling abundance is measured on such small plots and varies spatially. It's so spatially heterogeneous. And I just wonder how you think about balancing those abundance estimates against that kind of like sampling intensity sort of question.
Well, that's a great question. And I think part of this data exploration is figuring out, can we use the seedling abundance data that we're getting from FIA? Like, is it suitable for this type of research? And that's something we're still talking about and still exploring. Even when it comes to like the time, like how often the data is collected. So it's an ongoing challenge. So that's the best I have.
I think we have time for one more question. I see Margaret has her hand up.
Sure, yeah, there's some in the chat also.
Yes, there's a bunch in the chat.
Yeah, so maybe Ayanna, you can respond to those at your leisure. I guess I was wondering about the FIA seedling abundance data, are you taking everything from the year 2001 to the most recent FIA data that are available and just putting them all into one pool? And relative for any given location, like, do you have information about how recently fire happened? I think I saw you had a time since fire. Anyway, it's such a fire-driven species. I'm wondering how you're treating time. With the FIA seedling data.
Yes, so we took all the seedling abundance data from 2001 to 2020, 2020, sorry, what am I saying? To 2020. And we also devised a time since fire using the MTBS fire data. So we associated that with the plots and calculated the time since fire for that. I'm not, is there a second part to your question that I missed?
Yeah, I was just curious, like, because maybe a given plot might have been measured twice.
Yes.
Within that 20 years. And so then I guess if there's nothing that's time varying, I was thinking of climate data, but the only thing that would be time varying would be the time since fire.
Yes.
Everything else is only spatially varying, right? All the canopy structure, I guess, would be time varying too. So you just take like whatever the canopy structure is at the previous time that plot was. Yeah, I'm curious about how. Yeah.
So we take it at the time of measurement, and for each plot, we have a time since fire of first measurement and a time since fire of the second measurement, and we do our analysis based on those two.
Unfortunately, I'm going to have to cut this conversation short in the interest of time.
Awesome. Thank you.
Thank you. Okay, our next presenter, and unfortunately, I didn't have a chance to chat with him before, so I'll mess up pronouncing his name, is Yannek Käber. Is that good enough? And
yes.
Great. Yannek, you, I think, have many institutional affiliations, so originally, so I'm going to just say you can mention them, but you will be presenting on integrating empirical data with process models to infer forest demography, and it's very late where you are. So thank you for coming and presenting at this late hour.
Yeah, thank you for having me. And yeah, first of all, I want to clarify why there are so many institutions. So I did my PhD working with on questions regarding forest regeneration, and I will be working on a new project starting in January, and this is also where my talk will end. So first, I will we'll talk about some empirical models and insights of tree regeneration from various forest inventory data, all in Europe. And then also a study and some thoughts on inverse calibration of a process-based regeneration model, and then my new project I'm starting to work on next year. So first of all, I want to have a look at. I want to introduce how we can, or how I approach questions, and how I think forest structure, forest dynamics can be studied, understood, and also predicted. And so we have, on the one hand, we can choose the data models that are correlatives, quite simple and very easy to calibrate. And then on the other hand, we have the process models that allow us to also make use of mechanistic knowledge on processes, but those are rather complex and really difficult to calibrate or to connect to data. And yeah, so with process models in the context of forests, I speak of dynamic vegetation models. And for the data models, we have a range from structural equation models over generalized linear models and machine learning or deep learning models. So as I said, I'll start with some examples and insights from data models or GLMs. And yeah, so this is one study we did in when I was working in Zurich, Switzerland. And here we use different kinds of forest inventory data. So this is very heterogeneous data, and which consists of large plot inventories of a couple of hectares to 50 hectares, and then also systematic sampling grids of the National Forest Inventory in Switzerland and also of Northern Belgium. And here we fitted generalized linear models, mixed models to study the effect of stand density and climatic drivers. And we also differentiate between low shade tolerance species and high shade tolerance species. And what we found is that there's obviously a negative effect of stand density, which is to some degree modulated by a species shade tolerance. And at the same time, we find rather weak signals for temperature and water relations. And yeah, I didn't really explain. So the axis, just for your understanding. So here on the x-axis, you see the different inventories and the caliper measurement. So the diameter measurement threshold. And yeah, as I said, the prediction, the responses to ingrowth. Okay. And then I also did another study on forest regeneration, but with a strict focus on unmanaged forests. And unmanaged forests are not very common in Europe, so that those are very hard to find for historical reasons. And therefore, we have this Euphoria network, and where I compiled a huge data set from many different data sources to look into the topic of regeneration in these unmanaged European forests. And here the question we investigated was whether what's the role of positive and negative interactions for tree regeneration. So we again looked into climatic drivers, the seasonal water balance and temperature, but here we will only look into the water balance. And the response here, the modeled response here, is the mean of a negative binomial distribution. And the thick line shows the prediction for a dense forest, so with a high leaf area index, and the dashed line shows the prediction for a light forest, so with a low leaf area index. And what we found for some species with certain properties regarding drought and shade tolerance, that we find if there's climatic stress, so silver fir here, Abies alba, actually profits from a denser stand. So it can make use of positive interactions from surrounding trees. But on the other hand, these trees are affecting, the negative effect of these trees is much higher if the environmental or climatic conditions are more friendly for the species. And yeah, so we found these positive interactions, and we looked into this for different species with different drought and shade tolerances. And we also found that there's, to some degree, these differences can be explained by the species life history strategies and their tolerances to climatic stress. Okay, let's get back to this graph. So now we had a look at the data models, and now we have with the one downside of the data models is that those are not really suitable to extrapolate or to predict into environmental space where we do not have observations. So this makes it necessary to also look into process models, and regeneration is very difficult to capture, as the first talk also showed. It's really hard to get a good signal here, and it's even harder to model it in a process model. So this is why I was interested in looking into a very widely used forest gap model, at least in Europe. It's used for many purposes. And this model has different formulations for growth, mortality, and regeneration. And here we look especially into model structure and then species parameters. And our goal was to figure out which model structure or which species parameters can we infer from the data. And for this, we again use the euphoria data and to predict or to estimate in a Bayesian framework these parameters. And here you can see in the different panels for the different parameters, and also the different species on the y-axis. And for reference, those what I call trait-based values that are essentially related to Ellenberg-Zeiger values. Those are the model default parameters, and then we calibrated two model variants, so a simple and a more complex model. And so it's sometimes we match the trait-based approach and sometimes not. So I cannot really say what is better, but it's just that there's some room for interpretation and discussion when we look at these. But one thing we can see is that the credible intervals are much narrower for the light requirements, and still it's very hard to get a climatic signal. And this either means that there simply is no climatic signal and the within stand dynamics actually determine the regeneration and climate is not more important, or the model structure fails to actually capture these signals. Okay, so now for everyone who try to calibrate such process model, it's really difficult. So there's a lot of programming necessary and a lot of methodological things you can have to take care of. And this is where I developed, or me and my colleague Maximilian Pichler from the University of Regensburg, also in Germany, we developed as a side project this model, which we call FIN, which stands for Forest Informed Neural Networks. And here we want to bridge the gap between the process model and the data model. And so we get actually the best of both worlds. So this model will not be as good as the very as detailed as the best DVM, but also not an entire data model. So what is FIN? So FIN is basically a forest gap model. It simulates the demographic processes. It is a differential. It runs in discrete time steps, and so we have cohorts which represent trees. We have the environment as variables here, and these different processes. So it simulates on independent patches, and the competition is simulated by including height, and crowns are represented as thin layers on the top of the tree. And this model simulates classic patterns of forest succession. So this is just a random draw of an hypothetical species, and those trajectories are just to look whether it's plausible or not. Yeah, and the interesting thing about this model is that it is really designed for calibration. So it's just complex enough to say that it includes the basics of the basic properties of a forest, but it's quite easy to include data models. So and we can even replace an entire process by a data model, which can be a GLM or a neural network. And this is possible for all the processes, and it's very modular, and it's designed to be quite easy to use. So we really cover the whole range from a process model to a data model. And we implemented, so we have an interface in R where we want to make it accessible for different studies that want to address issues in specific issues or questions, but want to use also where it makes sense to use a dynamic model. And here, this is what the central function of this R package will look like. So we have here the observed data. This are standard, some demographic rates here, the growth, regeneration, and mortality, and some basic stand characteristics. We can also include climatic variables, and here those are now the central functions or functionalities of this fin function here, because here we can specify the relation of the environmental predictors to the process, and we are even able to define process the process or the mechanistic process specification, and how these variables are then used in a mechanistic model to simulate these different processes. And we hope that this offers a lot of opportunity for also specific studies. And the good thing is that the model runs really fast, so it's implemented in Torch. This means that if we have sufficient computing resources, we can add a large number of sites, but the runtime will only increase marginally to this.
One minute, Yannek.
1 minute, yeah. So I will skip this. So let's sum it up. Yeah, so what I learned from this forest inventory data is that regeneration can actually profit from dense stands under climatic stress, and I really felt the need to look into forest demography through the lens of a process model, and this is why I developed this model. And actually, I was just like I was already working on the FIA data because I find it quite tempting to calibrate the model on this, but I'm still struggling with cleaning the data and getting it in a format to actually calibrate it. So if someone knows someone who can maybe help me, or you're interested in collaborating on this, I'm happy if you get in touch with me. And yeah, so we will make this model probably available in a couple of months. So we're still working on it, and I'm now happy to answer some questions.
Unfortunately, we do not have time for questions right now, but Yannek, watch the chat and see if there's any questions, and maybe we'll have time at the end as well. So thank you very much. Next up, we have Kyle Rosenblad from UC Berkeley, I believe, who will be presenting on climate change, tree demography, and thermophilization. There's a mouthful for today. Western U.S. forests. So Kyle, take it away.
Thank you so much, and thank you all for being here. So yes, that's right. I'm finishing up my PhD at UC Berkeley, working with David Ackerley. Today, I'm going to present some results from a collaboration with Catherine Bayer from FIA, and basically, we were interested in using the most recent 10-year time step of FIA data to characterize sort of broad scale changes in forest community composition over that time step. Look at how that relates to changes in climate, particularly from a demographic perspective, and think about what all that might mean for future management. Before the details of all that, just want to make sure I acknowledge my wonderful funder, as well as some folks who provided very helpful advice on this work. Okay, awesome. Thank you. Sorry about that. So yes, was just acknowledging my funder and some folks who provided very helpful advice. Okay, so now I just want to use this cartoon to try and illustrate the motivation behind the work. So on the left here is just a very simple cartoon schematic of the process of thermophilization, which is that mouthful from the title. So the basic idea is the more we've been studying changes in community composition over time and looking at how they relate to changes in climate, the more we're finding that warming temperatures tend to produce a temporal increase in this thing that we might call a community temperature index. And the idea there is you just average across the sort of biogeographic affinity of each species, where if you broke it down into discrete categories, like I've done in this right side of the cartoon there, you might think of low temperature species, medium temperature species, and high temperature species. But of course, we can do it in any sort of more sophisticated way we want. But the idea is we're seeing more and more of these broad changes in community composition as the temperature warms, and other facets of climate like precip and things like that change as well. But the thing that we wanted to try and add on to this discussion about thermophilization is a little bit more of a demographically informed perspective. Because as you might imagine, that simple cartoon graph on the left could be produced by a whole wide range of different demographic scenarios, some of which would be a lot more worrisome than others from a management perspective. So just to illustrate that, on the right there, we've got sort of a before time point, a forest with some low temperature and medium temperature species at the top. And then that sort of bifurcates at the bottom down to two very different ways that we could produce a thermophilization scenario relative to the next time step. So on the right, we've got this sort of nice infilling of higher temperature species, some losses of lower temperature species. And depending on your management goals, that may or may not be concerning. But if you want a forest to persist in this location, then that would be relatively good news. Because the other way you can get a shift in community temperature index is just by total erosion of low temperature species with no sort of compensatory increase in high temperature species to stabilize biomass. And so this is the kind of thing we wanted to try and look under the hood and see which scenarios are driving thermophilization. So to outline the questions a little more formally, we want to know, is tree community temperature index increasing on pace with warming for the Western US? How do mortality, growth, and recruitment processes contribute to overall changes or overall thermophilization? And finally, what other sort of rate modifying factors like hydrology or disturbance might influence rates of thermophilization as well, either to accelerate it or perhaps to decelerate it? I wish I had more time to discuss the sort of motivation for each of these, but I'll just briefly run through our laundry list of things that we thought might matter in terms of influencing thermophilization rates. So perhaps most obvious, we would think that places that experience more warming should show more thermophilization. Places that dried out more, as represented by decreased precip or increased climatic water deficit, we thought might play a role as well. Topographic heat load is kind of an interesting variable. It basically just distinguishes cool north-facing hill slopes from hot and dry south-facing hill slopes. And we thought that might have an influence as well, perhaps due to increased vulnerability on the north slopes. And then disturbance events like wildfire or insect outbreaks, which unfortunately we don't have the sort of granular data to break down bark beetle species or anything like that. So for us, insects are kind of just one broad category of exposure event. Just quick details on the study domain. After some data filtering protocols, we wound up with about 11,000 FIA plots across the contiguous US, minus Wyoming for technical reasons that some of y'all might be familiar with. And for it was helpful, particularly in terms of showing fine scale topographic variation, to break things down at the subplot level. So we've got appropriate, hopefully appropriate random effects in our model structure to deal with that grouping structure. That left us with about 110 tree species in the data set, each of which we did a bunch of niche modeling to sort of characterize their biogeographic affinity, which then feeds into that thermophilization calculation I was talking about earlier, which, again, I don't have time for the details now, but I'd love to answer questions if anyone's curious. So we've got one 10-year time step with a before and an after FIA census for each of these plots, with the baseline census range of dates going between 2000 and 2008. And just a quick note on how we did this sort of demographic decomposition. This cartoon is just showing sort of how we analyze the data, either just to look at overall changes, which is what we're seeing on the left column there, where we've got, I should have called that T0 and 1, but a before and an after census where there's 2 species, red and blue. Some of them grow, some of those individual trees die, and some of them recruit. And then when you want to isolate the effect of each demographic process on thermophilization, the idea there is that, like, to take mortality as an example, we just said, let's have mortality be the only process that is allowed to operate between the baseline and follow-up censuses in our data. So we generated a whole different version of the data set where any changes in forest structure other than mortality were just sort of eliminated. So like growth was held constant, recruitment was ignored, and so forth. You can imagine how we sort of do analogous things to isolate the effect of growth and the effect of recruitment on community temperature index over time. Okay, so just to summarize briefly what we found here, this top panel is just showing us things we all probably, for folks who work in the Western US, already know, which is that it got a lot warmer for most of the Western US over the recent decadal time step. Things got a lot drier as well. That's what our second and third panels are showing us. On the bottom here, this is showing the forest community composition responses. So on the right, on the left, that's just a map showing that there was certainly some heterogeneity, but on balance, thermophilization was the trend. There's more red there than blue. Most forest plots, on average, increased in community temperature index. And this kind of funky scatter plot on the right is just showing us the same thing. It's showing us on the x-axis the rate of warming, on the y-axis the rate of thermophilization. The red dot there is the sort of data set wide mean. So on average, it was about a 0.04 degrees C per year rate of warming and a much smaller rate of thermophilization. And that gray line there is the 1-to-1 line, which is what we would expect to see if thermophilization was occurring perfectly on pace with the rate of warming. And so the fact that the red is far below that gray line is an indication that there is a lag there, as some other studies, particularly sort of understory studies, have been showing. But that's not something that's been explored quite so much for the overstory like we're seeing here. This figure has an awful lot going on, so I'm just going to pick out some key parts for the overall story here. These are the effect sizes from a bigger and more complicated analysis. But the y-axis here is just showing us for a whole bunch of variables the effect size in our model in terms of the effect on thermophilization rates. And these four sort of chunks of little violin shapes here are for the model where we allowed all processes to happen as they do in the real data. And then for the three scenarios, growth, mortality, and recruitment, those are those alternate versions of the data set where we isolated one demographic process. Specifically with recruitment, that's recruitment into the 5 and above size class that's often used in FIA. So key points here, the powder blue is just showing us that overall there was thermophilization. When we look at the growth powder blue one, that's showing that growth did contribute to that overall thermophilization, as did mortality. But crucially here, recruitment is just showing absolutely no hint of an effect there. And this is true when we did a sensitivity analysis with the smaller microplot sapling data. And to the extent that the seedlings could really be analyzed in that way, we were seeing a sort of consistent story as well. So recruitment is really not part of the thermophilization story here. A couple other things to point out in terms of different facets of climate change. This sort of salmon color is showing us that warming played a role. And the gold and the green are showing us that in most cases, drying played a role as well, perhaps not super surprisingly. This dark purple is kind of interesting. This is showing us a negative effect of topographic heat load. So the basic idea there is that thermophilization is faster on cool north-facing hill slopes, and that's more than anything else due to differences in mortality. So that's basically saying that there's there's already a bias toward mortality of low temperature associated species, but that bias is even stronger on those north-facing hill slopes. There's interesting stories too here about wildfire and insects that I don't have too much time to get into, but just the effect size for the insect outbreaks was massive compared to other things, still unpacking the reasons behind that, especially the question of whether that's just kind of an indicator of finer scale drought that our macroclimate data didn't pick up, versus an indication of true causality where the bark beetles were really doing something that the drought wasn't already doing. Okay. And so I just want to take a step back from the that topography result I mentioned a little while ago and just sort of illustrate what this might mean for landscape scale conservation. So I've got one of these little cartoons again here, where this time the land surface is color coded by topographic heat load. So the red on the right there is the equator facing, or for us in North America, the south facing hill slope. And over here on the left, that's the north facing hill slope, where topographic heat load is lower. And so in this landscape, at the sort of beginning time point, we've got some species habitat filtering, where the higher temperature species are on the hot slope and the lower are on the cool slope. But what's going on here is a little bit of a difference in the demographic story between those hill slopes. So at the beginning, we've got this nice sort of climate-driven beta diversity that increases the overall landscape scale diversity of tree species. But because that north-facing hill slope, which was the last refuge for this low temperature sort of fir-looking species here, because there's really strong losses of those most vulnerable species, what we're seeing is a sort of homogenization across the landscape if this process continues, whereby these sort of last strongholds of low temperature species on north slopes are just not going to be viable anymore. So to summarize, we're seeing a lag of roughly tenfold in the thermophilization rate relative to observed warming rates. And that is driven much more strongly by mortality and growth, with basically no role of recruitment. Thermophilization is increasing with rates of warming and drying, and insect attacks were showing a big effect size there as well. And thermophilization is eroding landscape scale biodiversity through those losses of those most vulnerable low temperature taxa on north-facing hill slopes in a given landscape. And as we're seeing with that lack of recruitment-driven thermophilization, it looks like warmer-associated species, at least thus far, have been slow to fill in at this sort of fine spatial scale that we're analyzing things at with FIA plots here. And I think that's an interesting contrast to some of the sort of broader scale work that says things like, oh, well, species are migrating into new climate zones at a rate that suggests they might do okay. And this is just sort of highlighting that that's potentially good news, but a different thing than being able to assume that those species are necessarily going to fill in forests at a finer scale at a faster rate. So just want to point out there's a paper out on this work, came out last year that y'all can consult for further detail on anything we don't get to in questions. But with whatever time remains, I am happy to answer some.
Cool. Thanks, Kyle. Unfortunately, we don't have any time for questions.
Of course.
Yeah, keep an eye on the chat,
Great.
and hopefully we might have a couple minutes at the end.
Great. Thank you.
All right. Wow, I just noticed my head is cut off. All right, so next we have Donnie Perret. He is a ORISE postdoctoral fellow with the Pacific Northwest Research Station and the organizer and really the motivator of this session. And so he's going to be talking about demographic pathways of subalpine forest resilience to global change. Take it away, Donnie.
All right, cool. Thanks, Harold. Yeah, like Harold said, I'm Donnie. Work as an ORISE postdoc at the PNW station with Harold and with Dave, who've been our fearless moderators here. And I just wanted to say before I launched into my talk, thanks so much for everyone presenting in this session, for agreeing to share your work. It's awesome hearing it all. And to everybody who's listening, I hope that this leads to some fun and productive conversations, both during the session and afterward. So I'm going to share work that we've been doing over the last couple of years, focusing on how subalpine forests respond to this confluence of global change drivers. And we started thinking about this work because subalpine forests, in particular, these high elevation systems, are experiencing a lot of rapid change. So temperature increases, decreases in snowpack, drought, high rates of insect and disease damage, unprecedented change in wildfire regimes in terms of both severity and extent. And the evidence is mixed about whether we should expect widespread transformation through these forest types, or if they will be resilient in certain circumstances. So a couple years ago, we started thinking about this, and we used subalpine fir as an initial point of attack into this system and as a case study. This is a paper I presented at the last FIA Science Symposium, and the work I'm going to talk about today builds on this, and hopefully will be out early next year for you all to read. So in this work, focusing on subalpine fir, we used FIA remeasurement data to estimate population trends across the whole distribution of the species in the Western US. And in particular, we estimated trends in basal area and abundance. And I'm going to show you why it's important that we think about both of those in conjunction. So imagine a situation where basal area is decreasing, but abundance is increasing. So when might that happen? Well, when large trees are replaced by small trees, right? When the population has a lot of turnover, as we might see following a disturbance event or something similar. If we flip those rates, so basal area increases and abundance decreases, that might just indicate some normal stand development processes. And if we fill in the rest of this little matrix of possibilities, if both are increasing, that stand will be actively densifying. And if they both decrease, that might indicate a true population decline. So for subalpine fir, between 2000 and 2019, we estimated, or rather we found declines in both basal area and abundance concurrently across a substantial portion of subalpine fir's distribution in the Western US. So that finding left us wondering what that means for subalpine forests more generally. How do we move from this single species perspective to learn something more about the community itself? So in thinking about how to do that, we took some inspiration from this excellent recent paper by Rupert Seidel and Monica Turner. And in this paper, they present a really nice conceptual framework for thinking about how forests might reorganize following a disturbance event. So they think about changes in structure and composition. Now, if both of those stay the same following some event, that's a resilient system. If they both change following an event, then the system's been replaced by something else, or they can change independently through restructuring or reassembly. Okay, so we're going to apply this to our population trends. If we move from this single species framework on the left to the simplest iteration of a community, so just two species, there are 16 ways that these four pathways combine, right? And what I'd like to suggest is that these reorganization trajectories that Seidel and Turner present map onto this space pretty cleanly. So if both species in our community have positive population trajectories, that would indicate that that system's been resilient to what it's been experiencing. If one or the other has undergone significant turnover of individuals, replacing large trees with small trees, that would indicate restructuring as the size distribution in the population has changed. If their trajectories are mismatched, that'll lead to reassembly as one species dominates over the other. And if they're both in decline, then that system will become replaced by something else. So this is the kind of framework we're putting together. There's a really nice two-species system in subalpine forests that we can use to build this out further and dig in here. And that's the Subalpine Fir Engelmann Spruce Association. So these two species form co-dominant stands across most of their joint distribution in the Western US. Where you find one, you tend to find the other. Prior work has shown that they're able to maintain that co-dominance and that coexistence through this balance of demographic rates and strategies. So subalpine fir for a tree has a faster demographic strategy, more regeneration, lots of seedlings, they grow pretty quick, but they have relatively low survival compared to Engelmann spruce, which has a slower demographic strategy, less regeneration. They're not shade tolerant. They grow more slowly, but they tend to have higher survival. So these two, this forms a balance, right, where they're able to maintain stable coexistence in their co-dominant stance. However, we also know that when climatic conditions were different in the past, this wasn't the case. So there's paleoecological evidence that suggests that at the last glacial maximum, these two species had pretty different refugia and pretty different distributions. And so something we might be interested in knowing is, as conditions continue to change in the future, could that demographic mechanism allowing that coexistence break or change in some way? So how will changing conditions impact the coexistence of these two species? So the way we're going to get at this is by connecting these reorganization trajectories that I presented earlier to these demographic mechanisms of coexistence and their responses to global change drivers, and then assess how that changes in future climate and disturbance scenarios. So this is kind of a two-step process. The first is we build a series of demographic models using FIA remeasurement data. So we modeled mortality, regeneration, so seedling presence, and recruitment, so new sapling recruit presence. As a function of a bunch of things, we're going to focus on climate. So these were mean annual temperature and climatic water deficit normals and anomalies, and disturbance. And here we focused on fire extent and severity. We then used those demographic rate estimates to predict the joint species trajectory observed on the landscape. I'll show you how we did that. I don't have time to talk about this demographic modeling step. There are a lot of interesting results and responses that we pulled out there. I'm happy to talk about them more later. But I'm going to focus on the second step, showing you how we combined these demographic estimates into this community framework. Okay, so the first thing to do is to estimate population trends the same way we did with our subalpine fir work for both species, and then combine them into this joint trajectory classification here. So this is what that looked like between 2000, 2009, and 2010, 2019 inventories. So we do have substantial areas of resilience, so positive population trajectories of both species across the distribution. Center here in the Northern Rockies. We see a lot of replacement and reassembly in other regions. So down in the Southern Rockies, where there have been substantial insect outbreaks. What we'd like to do is build a model that predicts these observed trajectories as a function of the demographic rates we estimate within each ecoregion subsection there. So we did this using a multi-newly generalized linear model. It's a classification model. This was a new trick for me. The model works pretty well. So we have a classification accuracy across all four outcomes of 60% or so. That's better for some outcomes. So we're really good at predicting resilience with accuracy of more than 75%. But some of the other pathways like restructuring or reassembly are more difficult. Okay, so I'm going to show you how varying demographic rates impacts what the model predicted as the joint trajectory we should expect. So here we have the probability of mortality for subalpine fir on the x-axis, and the same for Engelmann spruce on the y. And I'm going to color in this mortality space according to the predicted joint community trajectory. So this is what this looks like. So we predict resilience only under really low levels of mortality for both species. Replacement, on the other hand, can occur at any level of Engelmann spruce mortality, but requires high levels of subalpine fir mortality. So we see similar asymmetries if we look at our other vital rates, so regeneration and recruitment here. And in general, let's say that these asymmetric responses reflect the different demographic strategies I described earlier for the two species. And what we want to know is how the outcomes that we're predicting will change as global change drivers force the demographic responses of the two species in potentially different directions. So how do these trajectories change as climate and disturbance change? Okay, so to show you how we went about assessing that, I'm going to summarize these trajectories we estimated in the map into this bar chart on the left. So this shows the percent of spruce fir forest area that falls within each category, which is down at the bottom. And we forced our underlying demographic models using a couple different scenarios and then carried those through into our classification model. So we looked at two different time steps, 2040, 2050, and 2070, 2080 under the most kind of what seems like the most likely future emissions scenario. Using two disturbance scenarios. So a high disturbance scenario, that's 20% area burned at 90% mortality, and a low disturbance scenario, 5% burned at 50% mortality. So when we carry that all through, this is what those predictions look like. So in 2040, 2050, and 2070, 2080. And what we see when we look at this is that disturbance has a larger impact on the distribution of these trajectories than projected climate change does. Now, I see that as actually a kind of encouraging result, in that we can do something to influence the amount of disturbance on the landscape. We can't do much to influence the warming trajectories we'll be on, right? Things like fuel reduction treatments prescribed in cultural burning, wildfire land use can all work together to reduce severity and extent of wildfire on the landscape, even in these sorts of systems. So given that we have limited resources to do those things, we'd like to know where we can make the biggest difference. And so we can map this out. We can map the change of probability of resilience as we move from high to low scenarios here. And this is what that looks like. So in some regions, moving from this high to low disturbance scenario increases the probability of spruce fir resilience by more than 75%. In particular, we see that in the Northern Rockies. And so what I suggest is that we have an opportunity for high impact management interventions in that area to benefit spruce fir forests. Now, there's a lot more to dig into here, but I'll wrap up by saying this is where we started, thinking about how these forests respond to drivers, and we address that by connecting these global change drivers to the demographic rates they influence, then linking those to these community pathways, thinking about how communities might reorganize, and then use a scenario-driven approach to say something about management prioritization going forward. And so what I hope as we move forward with this work is that we can continue developing this framework and analytical pathway to link from these individual tree stand level effects all the way through to landscape level change and management prioritization. So thanks to everybody who contributed to this work. And I can see Harold, which means I need to stop talking. So hopefully there's time for questions. If not now, then later.
I'm a brutal clock master, Donnie. Yeah, we're gonna move right around. Thank you, Donnie. I'm expecting some comments in the chat there. We're gonna move right along. So next up, our next presenter is Adam Noel. I hope I'm pronouncing that right. Sweet, from the U.S. Geological Survey, and he's going to be presenting about ecological transformation in Pinyon-Juniper woodlands in the southwestern U.S. So take it away, Adam.
Awesome. Thank you for the intro, Harold. Just getting my screen set up here. Awesome. Hopefully you all can see my slides. As Harold said, my name is Adam, and I'm a postdoc right now with the USGS Southwest Biological Science Center. So I'm working with John Bradford down there. And on this project, I had two awesome co-authors, Bob Shriver with the University of Nevada, Reno, and Shelley Crosby with Conservation Science Partners. And I believe she's now with the Forest Service in Fort Collins. So this project was titled, Where Can Managers Effectively Resist Climate-Driven Ecological Transformation in Pinyon-Juniper Woodlands of the U.S. Southwest? This project really had two main goals, which were just to ask how and where is projected climate change expected to alter the population growth of PJ species, and where can reducing woodland density serve as an effective strategy to resist projected population declines in some of these climate-sensitive species. So why do we care about PJ ecosystems? While they span a huge part of our country, they span about 100 million acres of the Western US, extending from Canada down to Mexico. They're really ecologically and culturally significant. People who have been living on this landscape have been interacting with these ecosystems for thousands of years. And they're also a really important dry niche for many biota across the landscape, many obligate and semi-obligate species. So it'd be really great if you could figure out, what is driving these species under climate change and find some more mechanisms to help preserve them. So these trees are drought specialists. They're very naturally tolerant to dry conditions and heat stress, but there still are drought conditions that come across the landscape that are harsh enough to cause mortality events. And we're all aware that future climate may be more dry or more hot. So mortality in these woodlands, Pinyon-Juniper, trees commonly co-occur. Mortality in these woodlands is highly variable. From a previous study in the early 2000s, they observed about 97% pinyon death and less than 0.5% of juniper death. And more recently, there's been a mortality event going on in northern Arizona where we're seeing a majority junipers dying off, but pinyons are relatively unaffected. So these species have slightly different responses to drought conditions, and it would be good to have a better perspective of how climate change is driving some of these demographic patterns. But trying to project pinyon-juniper dynamics can be complicated. Obviously, to track a population growth, you need to know both the mortality and recruitment. If you're going to have mortality increasing in the future, recruitment is going to need to keep pace to sustain that PJ woodland on the landscape. Otherwise, you're going to have that ecosystem transforming from potentially, PJ composition to other grassland or shrubland composition. And so when we're trying to project these PJ dynamics, mortality is relatively easy to measure. You can spot when a tree is dying off, pretty straightforward. But recruitment is much more difficult to infer. Often these inventories, as they're going through these field plots, they're going through and counting the seedlings in their regeneration subplot. But because these trees grow just so slowly, upon revisiting, the 5 to 10 year sampling interval, when you revisit and you see these counted seedlings, there's no real way to tell whether they're brand new seedlings or are they just recounts from a previous survey. So to try and project these population dynamics, we're using a new demographic model that was developed by Bob Schreiber in 2021. This model infers population mortality, recruitment, and growth rate. And it's a size-structured population model, so it takes into account the tree size and its capacity to, influence regeneration within the model. This model uses covariates like temperature, soil water availability as predicted from a gridded soil moisture model, and then tree density at the site. And so Bob constructed this demographic model. We've created the model fitted with current climate conditions across 2 sampling points with FIA observations. And then we fit the model with future climate projections to project pinyon-juniper population dynamics under future climate. And so we did this for 5 common pinyon-juniper species, Pinus edulis and Pinus monophila, and then Juniperus osteosperma, Juniperus monosperma, and Juniperus scopulorum. And we chose those 5 species because they're really widespread across the US and they have great abundance in the FIA sampling data frame. So I'm going to get into results here. I'm just going to start with this concept figure, kind of explain what we're looking at. In this concept figure, we're just going to be plotting FIA sites by their 2 rates. So you can see along the x-axis, if we were to say at a given site, we have high mortality rate and low recruitment rate, that population would be decreasing. So it'll be below this dashed line you see going across the figure, where lambda is equal to 1 and population growth is completely even. In contrast, if you were to have a low mortality rate and a high recruitment rate, we would say that that population is increasing, exists in the area above that dashed line. Population looks good in our eyes. So we fit this model, like I mentioned, under historic conditions and then ran it with future climate conditions. These future scenarios we used were a lot of different models from RCP 4.5 and RCP 8.5, some moderate and severe emissions. And then we projected out these population dynamics out to mid-century and end-century timeframes. And we went through this. We used a set of 13 different climate projections and then used the median results for everything that you're seeing here. So I'll just get right into some of our results when we're looking at Pinus edulis. Again, all of these dots that are on this plot, this concept figure, are FIA sites that are plotted by their historic mortality and recruitment rates. And so historically, 24% of these FIA sites are in decline. So they're below that dashed line going across this plot. Using that model, but plugging in future climate conditions and taking median results, we can see that the cloud of points starts to shift more towards that line. And I should have explained before, the gray points are historic, and you can see there are some isolines that are showing the density distribution of all these points. And so now the blue isolines are showing how that cloud of points is kind of shifted under future climate conditions. So this is under RCP 4.5 end century. We go from 24% in decline up to 42% of sites that have declining population growth. And then we can look at this under the more severe emission scenario, RCP 8.5, also end of century, and that number increases to just 44% of sites. So that's one of our species. We can look at another one that we saw was at risk, Juniperus monosperma. Historically, only 11% of sites had this less than 1 population growth rate. You can see this cloud of isolines is hovering pretty far above that dashed line. As we move to the future, these population growth rates projected under RCP 4.5 end century conditions, you can see the blue isolines indicating the cloud of points moving more towards that line, and fully 50% of sites are now have a lambda below 1 declining population growth rate. And if we switch to the more severe emission scenario, RCP 8.5, that number increases to 81% of sites that are now experiencing a negative population growth rate. So we tested 5 species. The 2 that I just talked about, Pinus edulis and Juniperus monosperma, were the 2 that we found to be at risk. We tested the other species, Pinus monophila, Juniperus osteosperma, and Juniperus scopulorum. They all do have slight population growth rate changes. You can kind of see some of the clouds of points shift a little bit, but neither their recruitment rate or mortality rate shifts enough to really move the site or many of the sites to a negative population growth rate. And then also you can see on this figure, these kind of density plots at the edge of the plot indicate how the cloud of points is moving. And you can see that for Pinus edulis, the increasing, or I should say decreasing population growth rate at many sites is driven more by mortality rate increasing. So you see the cloud of points shifting along the x-axis direction. Whereas for Juniperus monosperma, most of the increasing, or rather decreases in population growth rate is due to decreases in recruitment rate and only moderate changes in mortality rate. So that brings us to our next question that we're trying to answer. Can management have an effect on reducing the potential for pinyon-juniper decline? So for the sites that shift below this line where lambda is equal to 1, can management reduce some of those projected declines? And we wanted to ask this question in the context of the resist, accept, direct framework, which is a management framework that kind of puts a full scope of management options under consideration. So you can either resist transformation, take management actions that benefit the species that are currently on the landscape. You can accept that ecological transformation will happen, just recognize that vegetation composition may change, or you could try and direct that transformation, manage the vegetation towards a desired vegetative state. And so the resistance strategy that we're testing here is just a simple basal area reduction by 50% of any site. So all of these sites, we take the FIA reported basal area and just plug in half of that into our model to see how management, taking away 50% of the basal area, might impact these sites. So I'm going to go through those results now. On the left half of the screen, we can see, this would be the proportion of sites in yellow with declining growth rates with no management. But then if we look on the right, you can see of that 37% of sites that may shift to declining growth rates with management, with a 50% basal area reduction, we can keep about 15% of those sites with a positive population growth rate. And so as we go through the emissions and time scenario combinations, we can see that this percentage of sites that we can maintain with positive population growth rates varies across scenarios, and we can get fully up to about 18% of sites to maintain positive population growth rates with management, whereas with no management, almost 50% of the FIA sites we're projecting to end up with negative population growth rates. So we can do the same thing with Juniperus osteosperma. Look at the time scenario combinations and say, if we reduced basal area by 50%, how would this affect population growth rates? And again, we see across these time scenarios, a range between only 8 and 12% of sites could benefit from management for Juniperus monosperma and keep those population growth rates above 1. So just thinking about those results and in the context of the resist, accept, direct framework, we could say that all these sites that are classified in purple, they have positive population growth rates regardless of management. They're able to keep on chugging along and resist transformation passively. All the sites in blue are those that would maintain a positive population growth rate, but only when we reduce it to 50% of their current basal area. So those would also be sites that we can resist ecological transformation, but it's going to take some active management to keep those population growth rates positive. And then sites in yellow would be sites that we would say you either have to or suggest directing or accepting ecological change. Even with 50% basal area management, these sites still had a population growth rate that was below 1. And for a last result, we can kind of take a look at these from a geographic perspective and see where these FIA sites for, with both of these species in there are present. There's no real discernible geographic pattern, which suggests that there may be potential at a lot of these forested sites to try and keep these populations on the landscape. And so overall, our first question, how will climate change alter population growth rates? There's really 2 sensitive species that we identified, Pinus edulis and Juniper monosperma, whereas our other 3 were much less sensitive to climate change. And we asked, can reducing woodland density resist some declines? For Pinus edulis, between 15 and 18% of sites could benefit, whereas for Juniper monosperma, only between 8 and 12% of sites could benefit. And we put that in the management context of resist, accept, direct. And then just as a last slide for more to consider, we still have uncertainty in our results. There could be some maybe minor adjustments to FIA sampling protocols that would help really key these results in. Some things like improved links between plots and local conditions, so microplot like temperature and moisture conditions, larger sampling sizes for regeneration, and potentially more frequent remeasurements or tagged measurements, so we really know if the seedlings are brand new or if they've been there from a previous sampling inventory. So that's all I have. Really appreciate everyone being here this late in the day. For some of you, looks like I don't have time for questions, but thanks for being here.
Great. Thanks, Adam. Yeah, unfortunately, we, no time for questions, but yeah, take a look at the chat. And so our last presenter is Margaret Evans from the University of Arizona, who will be presenting on forest carbon estimation and forecasting with forest inventory and tree ring data. So take it away, Margaret.
Okay, so I feel like maybe the oddball out in that I'm thinking about forest demography from a carbon perspective. So yeah, let's dive in. So the sort of motivation for this is that this is a figure that Kelly Heilman showed earlier today. The forest carbon sink, the US forest carbon sink, has been declining since about 1990. So that's this line going up towards 0. And, why is that happening? Well, it could be in the eastern US, this sort of natural recovery from, that old field succession process, stand filling process, endogenous demography. It could be reduced growth caused by climate stress. It could be increased mortality, increased disturbances, and native and non-native forest pests, land cover change. My money is on all of these combined together and in different degrees in different regions of the U.S. And really the point is that to parse these things apart and, get numbers on what part of it is driven by each of these, we need to, sort of better quantify and have improved temporal and spatial resolution of the forest carbon sink, these past trends, better understanding to make better predictions about the future for these sort of reduced emissions goals that we have at a national scale. And so there are 2 things that I want to set up at the beginning in terms of the perspective of kind of the way that we think about things in my group and the work that we do. One of them is sort of an ecological forecasting approach. And this has been inspired by Mike Dietz and a number of different publications, his book, papers that he's published, et cetera. And really the idea here is that we want to do these sort of repeated near-term predictions, build our models based on data, include all these different processes, make predictions about forest carbon dynamics, and then forest inventory, including tree rings, is really ideally set up to make a comparison between what our model predicted things are going to look like next year's data. And then those next year's data come in, and we compare the model predictions against observations and hopefully improve our model skill by looking at the mismatch between the predictions and the observations. The second thing that I want to sort of emphasize is this demographic perspective on forest carbon. So here's a yet another sort of stock and flux diagram of forest carbon, forest ecosystem and carbon dynamics in a forest ecosystem, and the relationship between the land surface and the atmosphere in terms of CO2. But what we're emphasizing here, this figure was made by Kelly Heilman. Here, so there's 6 carbon pools, forest carbon pools that are represented here. So above ground live trees, and then the below ground component of live trees, we have litter, down dead wood, soil organic carbon, and then we have standing dead carbon. So 6 different pools. The focus of this talk is just on the above ground live carbon and then the standing dead tree carbon. And in particular, what this figure emphasizes with these sort of green arrows is demographic or sort of carbon fluxes that are really demographic processes. So we have growth is above ground biomass increment. And then we have mortality is transfer from the standing live tree carbon to the standing dead tree carbon pool. And so what we're going to do here is we're going to use both the National Forest Inventory field observations and tree rings to quantify growth and the climate sensitivity of that process. And then we'll simulate stand level live tree carbon and standing dead carbon. So these 2 processes together are the 2 demographic processes that we're going to focus on in this talk. And a very brief outline for the talk. I'm just going to show one slide of work that's already published where we did this tree level forecasting of the impact of climate change on individual tree growth rates for one species, Pinus ponderosa, in the state of Arizona. And then I'll spend most of the talk talking about what we've been doing to scale that up, both at in the sense of a biological scale, so going from just individual trees to the whole forest stand above ground live tree carbon and the transfer of that to standing dead carbon, and also scaling up regionally from just the state of Arizona to the whole interior west wide. So as I said, just one slide on this is published work already. The lead author, Kelly Heilman, is now a postdoc with the Forest Service through ORAW, and she's working in the Forest Carbon Group in the Northern Research Station with Grant Domke. So this is the work that came out in 2022, and as I said, it just focuses on ponderosa pine in Arizona based on 515 trees for which we had increment core data, and then forecasts the growth increment. And then also this is looking at the whole pool of the 500 trees. And then this is the forecast for a single tree in terms of diameter, just the size of the tree. So the forecast with climate change, you get a 22% decline in radial growth increment by the end of the century under RCP 2.6, and other numbers that are in the paper that you can check out. So what I want to talk about today is the sort of focus on this regional scale analysis that looks at forest stand scale above ground biomass increment. And so it's the foundation of this analysis is the same Bayesian state space model of tree growth that we used in the study focused on Arizona, the published work. So the state space model is the way that we fuse together the tree ring and the diameter measurements. The state variable is the diameter of the tree. So we feed into the model two kinds of observations, the diameter tape measurements, and then the measurements of radial growth increments from the tree ring time series. And then the model itself is a dynamic linear model, so that we're keeping track of the state variable through time. So diameter at time t plus increment leads to diameter at time t plus 1. So it's just a difference equation that governs the time evolution, the growth through time of the tree. And then increment is modeled as a function of the size of the diameter, competitive conditions, climate conditions, etc. So that's the part that looks like a sort of a normal linear regression model. The regional scale analysis involves 675 FIA plots from which we have increment core data to work with. So that means there's about a little more than 1,000 trees where we have an increment core that was collected somewhere between the mid-80s and the mid-90s. And then we have somewhere between 1, 2, or 3 diameter measurements of those trees. And then we have a much larger pool of trees on those same plots that only have diameter measurements. So those are the data that go into the model. There's a, so this is the sort of more complex representation of that model, the full kind of regression model that's involved in it. And then here's some validation statistics, just looking at R-squared values for in-sample increments in the black. So the R-squared there is very high. Held out increments, the R-squared is a little bit lower there. And then in-sample diameter, the R-squared is very high there as well. And then the other thing I want to just tell you about from this state space model at the regional scale, the main ecological effects, these are posterior estimates, the posterior distributions of the regression model coefficients. So we see a really strong positive effect of mean annual precipitation. So trees that are found at wetter places, they grow faster. There's a negative effect of tree size. There is a sort of broad negative effect of stand density index. And then we see a negative effect of time varying temperature. So maximum temperature and how it varies from one year to the next. And then a positive effect of time varying water year precipitation. And I won't go into any of the rest of the effects here. So all of what I just talked about is the sort of this first step of state space model fitting and validation. And so now I'm going to talk through the rest of the steps involved in scaling this up and forecasting landscape level forest carbon across the region. So the first thing that happens is we go from the 600 plots where we had increment cores, we lose some of those plots. Some of them don't get forecasted for various reasons, missing data, or the importance of ponderosa pine in the plot is not high enough on the threshold that we set. We wanted to work with just ponderosa pine dominated plots. So the first thing that has to happen is that the trees in those plots have to get sort of forecasted forward. They have to be stepped forward in terms of their growth up to the common year of 2001. And so we use the growth model, that Bayesian state space model, to grow the trees forward. And then we go up here to the sort of starting at the year 2001. We're forecasting, we're simulating growth and mortality forward. And that's this box here. So again, we're using that state space model, the regression coefficients, to grow trees forward in the simulation. And then they're also subject to mortality, because if you don't subject them to mortality, you know, you just have carbon just gets larger and larger and larger. And so this opens up a whole can of worms about choices that you have to make about, okay, what is mortality going to look like in the future? So that's what I want to spend the next couple of slides showing you, is just kind of walking through, opening up that can of worms a little bit and showing you what kinds of decisions we made. And happy to take criticism and feedback about that. So inside this little blue box, the mortality and climate change scenarios, there are two mechanisms of mortality that we implemented. The first one is density-dependent mortality, i.e. the -3 Helps self-thinning law that's well known in forestry and in plant ecology. And so this is applied at the subplot level. It happens whenever the subplot stand density index exceeds 60% of the max SDI that is species specific for ponderosa pine. And the equations are drawn straight out of the way that FVS works, the forest vegetation simulator. So we have this kind of baseline background mortality that's the stand thinning process. And then in addition, we added a growth-based mortality that's applied at the tree scale. So this is a mortality probability that increases if you're not growing so well. We're taking a tree that's not growing well to be an indication that it has an increased risk of mortality. And the rate at which that risk increases is both size dependent and then growth rate dependent, and that is fitted to FIA data. And it's not the tree ring data; it's the DBH data. So the 10-year remeasurement data are used to build this logistic regression and establish the relationship between the risk of mortality, tree size, and the rate at which the tree is growing. Okay, and so then background mortality is in our simulation moving forward. We tried to make sure that our background mortality rate before we apply climate change is in the ballpark of the observed rate of mortality for these same plots over the last 20 years, from 2001 to 2021. So it's a sort of way of using the near past as a way to calibrate our simulated mortality. And so that, so I'm going to just show you, this is the histogram of the per year, per tree mortality rate in these 500 and something plots that were simulated forward. And so you see this histogram, this distribution. We're calling 1% the sort of the value that's kind of normal as a background rate of per tree per year mortality. And so this is what that per year per tree mortality rate looks like in the simulation for actually the first 10 years of the simulation.
Hey, Margaret, just letting you know.
Sorry, what was that?
Just letting you know, you might want to wrap it up.
Oh gosh. Okay. All right. So yeah. So we applied climate change to mortality by ramping up the density-dependent mortality or doubling the growth-based mortality probability. We end up with 5 different scenarios, sort of a control treatment, one where climate change only affects growth, one where climate change affects growth plus the density-dependent mortality, and then one where climate change affects growth plus the growth-dependent mortality, and then one where and this is the one that I'm going to show you in the next figure, which is the sort of the full model where climate change affects both growth and density dependent and growth dependent mortality. Okay, so the rest of this, that was all in this little tiny blue box here, the can of worms. And then we just scale it up with allometric equations and then FIA population tables to get to landscape level across the region live tree carbon and standing dead tree carbon. This is my second to last figure, I'm pretty sure. So this is the results figure. So if we compare the simulation that has climate change impacts on both growth and both forms of survival or more mechanisms of mortality against the no climate change scenario, this is what the stocks look like. So live tree carbon and dead tree carbon. And if you look at the difference between these two, that's what we're showing in this B panel over here. And so climate change reduces live tree stock by 29% compared to the counterfactual of no climate change. And it increases dead tree carbon by 25% compared to the no climate change scenario. So take-home messages, combining tree ring and forest inventory data together allows us to make this climate sensitive forecast of tree growth. Scaling that up to above stand level, above ground biomass and biomass increment and carbon stocks and fluxes of live and dead trees means we have to simulate an additional demographic process. Here we did mortality and we left regeneration completely out of the picture. And I didn't get to show you this, but the reduced growth is actually a minor component of the changes in stocks that I just showed you, and really it's driven by the scenarios of mortality that were created. I showed this slide earlier today, so maybe I won't spend too much time on it, but we're working on creating annual resolution forest carbon estimation and forecasting through this forest time project with the carbon science group. And with that, I will stop.
Great. Thanks, Margaret.